Hi dear students, in this video we shall explore the structure and functions of ribosomes and Golgi apparatus, which is also known as Golgi bodies, lysosomes, peroxisomes, and glyoxysome. The cell contains many tiny granular structures known as ribosomes. Palade in 1955 was the first person to study them. Eukaryotic ribosomes are composed of an almost equal amount of RNA and protein. Hence they are ribonucleoproteins particles. The RNA present in the ribosome is called ribosomal RNA. They are synthesized in the nucleus and exist in two forms. Either freely dispersed in cytoplasm or attached with RER as tiny granules. Each eukaryotic ribosome consists of two subunits. The larger subunit sediments at 60 Svedberg. Svedberg is the unit used in ultracentrifugation, while smaller subunit sediments at 40 Svedberg. Subunits on attachment with each other form 80 Svedberg particle. This attachment is controlled by the presence of magnesium ions. The ribosomes are attached to messenger RNA through small ribosomal subunit. A group of ribosomes attached to mRNA is known as polysome. New ribosomes are assembled in the nucleolus of the nucleus, from where they are transported to the cytoplasm via the pores and nuclear membrane. The factory of ribosome is the nucleolus, while that of protein synthesis is the ribosomes. The Golgi apparatus, also known as the Golgi complex or Golgi body or parallel flattened, membrane-enclosed sacs-like organelles called cisternae, which play a central role in the endomembrane system or internal membrane-forming system, acting as a processing and packaging factory for proteins and lipids. Its structure is composed of flattened, membrane-enclosed sacs called cisternae. These cisternae are typically arranged in parallel stacks, often described as a stack of pancakes. While the number of cisternae can vary, a typical Golgi stack contains 4 to 8. The entire Golgi apparatus is supported by cytoplasmic microtubules. Structure of Golgi apparatus is divided into 3 main functional regions. Cis-Golgi network (CGN), cis face, forming face. This is the entry face of the Golgi, usually convex and oriented towards the endoplasmic reticulum Proteins and lipids from the enter the Golgi here, often via transport vesicles. The membranes of the cis face are generally thinner. Medial Golgi. This is the central region of the Golgi stack, where most of the metabolic activities and further modifications of proteins and lipids take place. Trans-Golgi network (TGN), trans face, maturing face. This is the exit face of the Golgi, usually concave and located farthest from the ER. From here, processed and sorted molecules are packaged into vesicles for transport to their final destinations. Associated with the cisternae are numerous vesicles that bud off and fuse, facilitating the transport of molecules within and out of the Golgi. Functions of the Golgi apparatus. The Golgi apparatus performs several crucial functions, primarily involved in modifying, sorting, and packaging macromolecules for secretion or delivery to other organelles. Protein and lipid modification. Glycosylation. One of the most significant modifications is the addition or modification of carbohydrate chains glycans to proteins, forming glycoproteins, and lipids, forming glycolipids. This is critical for proper protein folding, stability, and cell-cell recognition. Phosphorylation and sulfation. Addition of phosphate groups and sulfate groups to specific proteins and lipids are known as phosphorylation and sulfation respectively. Enzymatic processing. Various enzymes within the different Golgi compartments carry out a range of biochemical reactions that further process the incoming cargo. Transport. Golgi apparatus transports sorted and packaged vesicles. Secretion. Proteins and lipids destined for secretion outside the cell, such as digestive enzymes, are packaged into secretory vesicles that fuse with the plasma membrane, releasing their contents. Plasma membrane. Components intended for the cell's outer membrane are incorporated into vesicles that fuse with the plasma membrane. Lysosomes. Hydrolytic enzymes destined for lysosomes, which break down waste, are tagged with mannose 6-phosphate in the Golgi and packaged into specific vesicles that fuse with lysosomes. Lysosome formation. In animal cells, the Golgi apparatus is essential for the formation of primary lysosomes, which contain digestive enzymes. Cell wall synthesis. In plant cells, the Golgi apparatus is responsible for synthesizing complex polysaccharides, which are crucial components of the cell wall. In essence, the Golgi apparatus is like the cell's post office, receiving, processing, organizing, and dispatching cellular mail to its correct address.